Okay, so hello everybody. My name is Fabricio, and I welcome you to today's webinar, Measuring and Modeling Electroconductive and Quantum Properties of Cytoskeletal Protein Filaments Exposed to Electromagnetic Fields. This webinar is brought to you by the Scientific Computing Program at the Fiocruz Foundation at Rio de Janeiro, Brazil, and the Global Health Network Latin America and the Caribbean. Fiocruz is a prominent public health institution in Brazil, founded in 1900 and affiliated with the Brazilian Ministry of Health. It is responsible for various public health activities, including research, education, the production of vaccines and other biopharmaceuticals, and health policy development. The Scientific Computing Program from Fiocruz is dedicated to progressing and applying innovative computational, mathematical, and statistical methods in bioscience research, focusing on studies developed at Fiocruz. Okay, as I told you, my name is Fabricio Alves, and I am a researcher in public health at Fiocruz, Oswaldo Cruz Foundation, and a permanent faculty member of the graduate program in computational and systems biology at Fiocruz. And today, I'm delighted to be joined by Dr. Jack Tuszynski, a professor at the University of Alberta in Canada and the Polytechnic de Turin in Italy. Okay. A quick note on the agenda for this session today, okay? First, there will be welcome and introductions, and then we will have Jack's talk. And after Jack's talk, we will have an interactive session with panel discussion and Q&A. So participants can raise their hands after the presentation or post questions in the Q&A Q&A feature throughout the webinar. And in the end, we will have some concluding remarks. Okay, housekeeping. Before we begin, we will run through some housekeeping. Firstly, of course, this webinar will be held in English. This session is being recorded. In the coming weeks, we will share the session recording on the Global Health Network platform at the Fiocruz and Latin America and the Caribbean hubs, so you can review and share the materials. You can find these materials using the link we will share in the chat. Due to the number of expected participants, your video camera and microphone have been turned off. Please use the chat feature to report any technical issues you might be experiencing. The Global Health Network Fiocruz Hub team will assist you with those issues. To interact with the panel, please use the Q&A feature to post your questions and comments. You may do so anonymously. And there will be an opportunity at the end of the session to ask questions. Please raise your hand and open your microphone. We'll try to get through as many questions as possible. Finally, it's my pleasure and honor to introduce Professor Tuszynski. Jack Tuszynski is a fellow of the National Institute for Nanotechnology of Canada. He is an Allard Chair and Professor in Experimental Oncology in the Department of Oncology at the University of Alberta's Cross Cancer Institute and a professor in the Department of Physics at the same institution. He is also a professor at the Polytechnic di Torino, Italy. Professor Tuszynski received his master's in physics with distinction from the University of Poznan in Poland in 1980. He received his PhD in condensed matter physics from the University of Calgary in 1983. Jack, thank you very much again for accepting our invitation. You have the floor.
Thank you very much, Fabrizio. Thank you, everyone, for participating in this presentation, and thank you for inviting me to speak to you. I have very fond memories of my visit to Rio de Janeiro, to Fiocruz. It's a great place. And I also today will make a connection to another place in Brazil that is figuring in this topic, namely Syro-Lebanese Hospital in Sao Paulo that I visited more recently. And its pioneering program in cancer therapy will be mentioned today. So I'll now share the screen and start my presentation, which hopefully you can see now. If not, let me know. Okay, I'll go to the slide mode. Okay, here it is. So I will be speaking about things which connect between biology and physics and hopefully open new avenues for basic and applied research, as well as therapies in the future. Most of the biologists don't think about electric currents and electromagnetic interactions. Most of physicists don't think so much about cells and proteins, but here we'll combine the two areas of investigation. I will try to cover about 20 years of research in less than an hour, so it'll be about a tour de force. But hopefully you'll take home some messages of importance for the future of biology, cell biology in particular, and other fields related to it. I want to very quickly acknowledge my collaborators from the University of Alberta that you can see here, students and faculty members, Al Meldrum and Karthik Shankar in particular, in physics and engineering respectively, and also my collaborators in the second part of this project, Aristide Dogario from University of Central Florida, and Chris Carlson from Harvard, and Greg Scholes from Princeton. We had some fun doing experiments in the last few years, and they continue actually. So also I want to acknowledge the funding for this work from various agencies in the US and Canada, as well as some companies that are listed here. And I will just mention by name. Novocure is a pioneering company from Haifa, Israel, that is using electromagnetic therapy to treat brain cancer. And V-Light is a Canadian-based company that uses electromagnetic means of treating depression and some neurological diseases. And they've been kind enough to support our work. And the Templeton Foundation recently funded our work on the investigation of quantum effects, which I'll mention briefly. It's a huge topic, so I don't want to spend too much time on this because it's a little bit of the beaten track. Now, let's start with the main central piece of the research investigation in all of this, which is microtubules. Most of you probably know microtubules from introductory biology as protein filaments made up of tubulin dimers, which create these cylindrical structures that play crucial roles in cell biology in terms of providing cell shape, rigidity, morphology. And they are importantly absolutely necessary for cell division in all eukaryotic cells. And they also participate in cellular transport, as you can see here on the right-hand side of this panel, where kinesin is carrying a big cargo moving along a microtubule. That's an artist's rendition, of course. It's a computer simulation, but these things are happening in real time, in real life, both in dividing and non-dividing cells. We'll be focusing our attention on dividing cells, where, as I mentioned, microtubules shown here in green are necessary to provide force in chromosome segregation. And we'll come back to this picture from some experimental data that I'll provide you. So what's a microtubule? MT stands for microtubule. Its building block is a tubulin alpha-beta dimer, and it's spontaneously forming these cylindrical structures, 25 nanometers across in the outer diameter, 15 inner diameter hollow cylinders. They can be anywhere from 10 micrometers to millimeters long under laboratory conditions. Typically in the cells, they will span half the diameter of the cell, so tens of microns. What we will be looking at today is very different from what probably most of you are familiar with, namely how these structures respond to electrical stimulation and what their electrical and electromagnetic properties are. So this is kind of a different view of systems biology that Fabrizio mentioned. Systems biology in terms of electrical grids spanning cells, including neurons. And in bullets here on the left, I listed some of the properties that we investigated and published papers on those remarkable properties of microtubules in ionic solutions and in cells, namely their resistance, which is variable, capacitance as electrical elements. They act as voltage pulse transmission lines. They also have something mysterious, which is called memristance. They keep memory of the past current flows along their lengths. And then towards the end of this talk, I'll mention to you some other interesting phenomena. Delayed luminescence. They absorb electromagnetic energy and release it over relatively long periods of time. And then they also respond very strangely and interestingly to terahertz electromagnetic fields. Okay, so one more thing before we delve into it, which is probably known to biologists, not so much to physicists, that microtubules, among other functions associated with mitochondria and different in different stages of the cell cycle, they carry mitochondria through motor proteins. Here is the interface, mitochondria along microtubules moving a little bit, fluctuating. And here is a video of mitochondria in red during cell division, mitosis. And you will notice that, well, maybe it's not clear from the picture, but if you know cell biology, they actually dissociate from microtubules during mitosis. Okay, so that's potentially of importance later on when we discuss what happens to microtubules exposed to electromagnetic fields. One more thing that I wanted to mention, because Fiocruz is involved in virology research. We published a paper recently that shows by computer simulations and corroborates some experimental facts related to microtubule involvement in the invasion of cells, host cells, by viruses. In this case, COVID-19. The coronavirus enters the cell by hijacking microtubules. This is a cartoon of this situation with the viral particles getting into the cell, and then they move on microtubules to, in fact, hijack the centrosome. And you can see here on the left the coronavirus rendition, a graphical rendition with the spike protein decorating the surface, and then placed inside the centriole. As you can see here, the centriole has 9 triplets of microtubules, and the coronavirus fits perfectly inside the centriole, and the centriole, being the sort of command center of the cell, is now hijacked, and the virus can direct the cell's movement and activities this way. We've done electrostat, so this is the first connection to electricity. As you can see here, this is a map that shows spike proteins in terms of the electrostatic charges, red being negative, blue positive, and white neutral charge distributions. And then you have the one protofilament of microtubules inside the centriole, for example, where you see the red being the negative charge on the C termini, and it really corresponds physically to the positive charges on the spike protein. So there's a strong electrostatic attraction, which the spike protein can utilize and control the cell this way. So this is a paper that you can find, or if you're interested, please contact me. I can send you the paper which elaborates on this. Let's now move on to other properties. So we start with electrostatics, electric charges in the cell. Maybe it's not so well known, but there is, of course, a huge number of different charged particles, starting with ions, the most common being potassium, then sodium, inside all cells with different concentrations, but roughly given here in this table, and then chloride and calcium and magnesium in the order of importance. And then you have proteins, actin and tubulin being the most abundant highly charged proteins, especially tubulin. And on the left, you see here a cartoon borrowed from Jerry Pollack's work, which shows you the cell with all these charges organized, and the water molecules also organized by the charges. That is another topic for another day, but biological water, cellular water, is dipolar, organized in hexagonal lattices, and most of the water inside cells is not free, like tap water doesn't have all the degrees of freedom, but actually it is organized, which is important and largely unexplored. I'm collaborating right now with a group of Rumiana Tsenkova at the University of Kobe in Japan to find out how ordered water in cells congregates around microtubules, actin filaments, and such. And then you have this diagram, which is very important, showing you the transmembrane potential in millivolts MV. Maybe I'll, yeah, that transmembrane potential, which changes depending on the cell type. So there's high correlation between the transmembrane potential and the type of cell that you're investigating. And in particular, here you have the largely abolished transmembrane potential in cancer cells, very low values from -5 up to maybe -25 millivolts, as opposed to normal cells somewhere between -50 and -90 millivolts. So these things indicate something important, and I would like to also draw your attention to the work of Professor Mike Levin from Tufts University in Boston, who is specifically investigating the role of transmembrane potential on the cell differentiation processes, cell division, etc. So this is kind of a summary of electrophysiological hallmarks of cancer cells as opposed to normal cells, with a well-known difference, as I mentioned, in the transmembrane potential, as well as the differences in both the pH values outside the cells in extracellular matrix, which becomes highly acidic in the case of cancer cells, and the opposite transformation takes place in the intracellular space, where pH is increased for cancer cells and decreased for normal cells, with potential implications because mitochondria are not shown here, that's the nucleus, but if you look at mitochondria, the activity of mitochondria in producing the energy molecules, ATP in particular, is proportional to the transmembrane potential difference between the cytosol and the mitochondrial pH. So if you increase mitochondrial pH is higher than cytoplasmic. If you increase the cytoplasmic pH, the gradient becomes lower, so the activity of mitochondria decreases, which is called the Warburg effect, switch from oxidative phosphorylation to glycolysis. So that's one aspect. Another aspect, of course, is changing the transmembrane potential, and the pH values is activity of ions and ion channels, which is schematically shown here. We published a review paper just a year or 2 years ago, which really outlines in great detail all the distinguishing electrical properties of cancer cells. And of course, we stopped at the basic science questions. Today, I'll say a few words about what can be done therapeutically in this regard. And on top of that, I want to mention one more thing, which is that in normal cells, pH is not just different. It's lower inside the cells, but it's also heterogeneously distributed, as shown here in these slides. In cancer cells, it's homogeneous, and so the gradient of pH inside the cell is largely absent or diminished. Now, let's go back to microtubules and focus on their electrical properties. So microtubules and tubulin, the building block of microtubules, are absolute outliers in terms of the charges, net charge on the protein shown here. This is the histogram on the left of net charge on various proteins, all proteins which were characterized here. I don't know if this is, yeah, it's about 20-some thousand proteins, and the average would be around -5 or so. Microtubules have something between -30 and -50, depending on the isotype, this and etc. So, but really much larger, and the same applies to the dipole moment. As you can imagine, counter ions will neutralize proteins in the cell largely, but what's left is the dipole moment, and the dipole moment here for tubulin is in the range of about 2,500 up to maybe 4,000 dBi. DBi is the unit of the dipole moment. For comparison, water molecules have a dipole moment of around 2 dBi, if I remember correctly, but it's a single digit, maybe 3. I think it's 2. So this is 1,000 times greater than or more than the dipole moment of water. These are other proteins, again, in a histogram, and you can see here that very few proteins have comparable dipole moments to those of tubulin. So here we have a working hypothesis that we put together with Dr. Degari from University of Central Florida, that because of the major roles of microtubules and microtubule constructs that include centrioles, microtubule bundles in neurons, cilia, flagella, everything to do with cell motility, cell division. So they are critically important. Their functions must be somehow related to the incredible differences in their properties, electric and electromagnetic. This can be used by cells to transduce signals over long ranges and also fast. You know, there's nothing faster than electromagnetic fields in terms of propagating at the speed of light, as opposed to chemical signals, which are very slow, and even ionic currents are much slower on the order of meters per second, as opposed to 300,000 kilometers per second for light. So this is a largely unexplored area which challenges the central dogma of molecular biology, and it's underdeveloped and underinvestigated, and I think it needs to be put in the spotlight. this shows you here a computer simulation of the microtubule charge distribution in various orientations. This is the structure of microtubule. Red and blue is actually just alpha and beta tubulin monomers. Here it's actually charge maps where red is negative and blue positive. This was published more than 20 years ago by the Baker group from UCSD in PNAS, and shows you that that a microtubule may have something like 500,000 negative charges per length of 10 micrometer, even if 50% of them are compensated by counter ions, you still have 250,000 electric charges forming a very strong electric field, which I will show you later on from simulations is indeed significant. And if you translate it into the number of microtubules and mitotic spindles of human cells, this gives you a staggering number of 345 million electric charges in the entire spindle. And that can be compared to the nuclear charge of 20 million electric charges. So overshadows completely. You've seen some of that. This shows you a bit more in more detail charge distribution for different family of tubulin proteins here in brown. So tubulin proteins, which is several isotypes of alpha, beta, gamma, delta, epsilon. There's a lot of tubulins, about 20 different genes code for tubulin, each a different variant. So that's why there's a distribution here. And this is for the dipole moment. In both cases, these are clear outliers compared to the unique proteins in blue. And it also shows you something interesting, not just statics, but now dynamics. Dynamics of electric fields is measured by polarizability. That means how the dipole moment, the change in the dipole moment with respect to the change of the electric field applied externally. And we've published this paper here, which we'll come back to later, but also experimentally measuring the polarizability of tubulin in solution. And there are several modes of polarizability, including highly flexible and highly charged C-termini that respond to electric fields, as well as dipole, permanent dipole moments, and the counter ions, of course, which congregate around a tubulin, so they will be movable. And as they move under the influence of electric fields, the net dipole moment of the structure shifts. And finally, water molecules as well have dipole moments, and they will be responding to electric fields. So there's a lot of moving parts, so to speak, and that shows you the experimental data published in this paper in the laboratory of Dr. DeGario in Florida, shows you how polarizability, this is measured by this data of tubulin changes as a function of pH, and it also depends, it has a hysteretic behavior, so it depends on the change of the direction of pH going up or down. There's a slight difference, and also the dipole moment measured in these experiments in around 2000 dBi, and the charge is pH dependent. It's quite remarkable. So, so I think what I want you to remember from this is that these things are tunable by pH changes, and to even a greater degree by ionic concentrations, which so pH is of course proton concentration, it's an ion as well. I think I showed you that before, so I'll just skip it. So all these external fields that we'll be looking at may affect a lot of biological functions, including microtubule polymerization dynamics will be affected by external effects. So that gives us some idea about things like creating controllable devices, bioelectrical circuits, as well as interfering with the cell cycle. And here's a summary of different mechanisms of applied electromagnetic and mechanical fields at the level of single microtubules and microtubule networks. These things change. depending on the electromagnetic frequency applied, which is basically resonant responses to various frequencies. This table is incomplete and requires systematic studies. We just looked at several aspects, as you will see. So there are effects of the visible light, excitonic energy, changes, electronic, conformational, ionic, and even mechanical. So all of this is largely frequency dependent and tunable. Other groups worked on this to some degree. This is responsive microtubules to electric fields. You can see here gliding assays and reasonably low electric fields. They microtubules in solution align with these fields. And you can see forming of microtubule parallel bundles as a result of electric field application. These are static electric fields. And this becomes quite a bit more complex if you apply frequency dependent electric fields. These are the groups in the US, I think Michigan, and this is a Japanese group that also studied this. It's now almost 20 years ago. Now, when you increase the frequency of electric fields to megahertz and beyond, then very interesting structures are formed. The white ones are microtubules, and you can see here that they change the morphology of these bundles of microtubules depending on the frequency. And these authors, Upalapti from University of Michigan, identified several mechanisms of interactions, dielectrophoresis, electroosmotic flows, and thermal conduction, because there's some heating involved in the activation of ion movements and protein movements. So we proposed some possible conduction mechanisms involving microtubules, and it took a while. This is 2015, and I think it took us almost 10 years to dig deeper into these hypotheses. And the hypothesis number one is that ionic waves propagate along microtubules. Some of the ions condense around microtubules, are not movable, but some are movable. And so microtubules form some kind of a biological electrical wire, for the lack of a better word. And they can conduct both the proton concentrations, so create pH gradients, which we recently demonstrated is the case. Ionic, so typically potassium or sodium waves, as well as potential electronic conduction, which has not been published anywhere to my knowledge. We've done some experiments on dry microtubules and measure dry conductivity, which would be not ionic, therefore must be electronic. But we need to do more experiments on this to obtain high quality data. And that's, again, an old result which started my fascination with microtubule conductivity. Here we see a microtubule held by two micropipettes with electrodes. That was a tour de force achievement by Professor Horacio Cantiela, who was at Harvard at the time. He's now at University of Buenos Aires in Argentina. And he was able to create this sort of single microtubule measurement of its conductivity in solution. And you can see here some amazing results, which is the blue. In blue is the input voltage, and in red is the resulting electrical signal, which has a greater amplitude. So microtubule here was showing amplification of electric signals. So we called it the transistor-like behavior of a microtubule, and developed some theoretical models published a couple of years later, where we represented the microtubule and its unit tubulin as an electrical circuit element with resistors. So this is the direction of the ion flow. This will be the surface of a microtubule, and there is a resistor due to the the viscosity of the fluid. So ions have some resistance to its flow, both along and away from the surface. And then there is a capacitance, which is variable because they congregate, they're attracted to the surface, but there is a finite number of counter ions that can be absorbed on the surface. So that's the variable aspect of it. And then inductance, which is because the flows, due to natural geometry of microtubules being helical, can form a solenoid as well. So we estimated the values of these parameters, very low values of, so picofarad capacitance, mega ohms resistance, and picohenries, sorry, femtofarads and picohenries of inductance. And this is a bit more elaborate, a model which was published in Physical Review a few years later, where we also allowed for the dynamics of C-termini, which actually carry about 40% of the charge on tubulin. So they play an important role, but they are very flexible, fluctuate, and that could be causing ion flows in and out of the microtubule lumen, a bit like a battery. What I want you to also take out of this message here is that there's a very sizable potential difference across the thickness of microtubules, about 120 millivolts, which is comparable to the membrane potential. The thickness is 5 nanometers, again, comparable to the thickness of a membrane, cellular membrane. And that gives you the electric field across the surface of a microtubule, again, comparable to that of a membrane, about 2 million volts per meter, which is very large, which tells us that there is a huge potential for using it in terms of transport and shielding effects and things of this nature. So more recently, from about 2015, we've been doing experiments looking at the conductivity, measuring ionic conductivity of microtubules, and we discovered that this is quite amazing. This is the setup semiconducting device with a 4-probe system. That's the sample here with electric field distribution. You have microtubules in white, and we measure this over several years under various conditions. We also measured capacitance because, as I told you, microtubules attract ions and form a double layer. And now everything changes depending on ionic concentration, depending on protein concentration. This is shown here. As you can see here, images of microtubules that were measured in these experiments. Here you have more or less parallel, somewhat disconnected microtubules. Here you have a network of random microtubules, and here it's almost like a spaghetti situation with very high level of connectivity. And we measured this as a function of frequency and as a function of ionic concentration. So the results were surprising to us. First of all, we found some broad peaks at about 100 kilohertz frequency. Then we found that microtubules are more conductive than tubulin and more conductive than the solution itself at low ionic concentrations. So that was very interesting and published several years ago here. And then so at low ionic concentrations, microtubules increase conductance. So they become really conducting wires. And at sorry, I can't see this. At high ionic concentrations, actually, the opposite happens. That's kind of intriguing, that when you increase ionic concentrations, and the question is at what point, and they become resistive elements compared to the solution itself. So you can switch the role of microtubules from conductors to capacitors, because at high ionic concentrations, they are more capacitive than the solution and also than the free tubulin. So formation of microtubules from tubulin polymerization can be seen also as a transition from solid to solid, and a transition from conductor to capacitor. And both elements are present, but they change the relative importance. And these are it's a lot of information here. I don't want you to remember any of these numbers, but they actually, what I want to emphasize here is that these are the concentrations of tubulin. So we increased the tubulin concentration from 0.2 to micromolar to 10 times, 100 times, and yet the system's capacitance is roughly the same, and the same can be said about resistance. So that's something interesting. That means that the networks formed are neither parallel nor series from the electrical point of view. There's something to be investigated. We haven't really been able to create very accurate models of this, but it's in its own right an interesting finding. And by extrapolation, we found numbers for this values of R, C, and L, which are actually comparable to our earlier predictions from several years before. And then also something that we'll come back to in a second, namely, when you just look at the resonant frequency of an RLC circuit, you obtain something like this from these numbers, about 0.65 terahertz. And very shortly, I'll show you experimental data that actually support this prediction as well. So this is the highest conductivity of microtubules at low concentration, 20 Siemens per meter. The solution has about a 10 millisiemens per meter, so it can increase actually conductivity by a factor of 2,000. It's very significant. Then I'll just very quickly go through some other experimental data. This is intriguing because at the value of pH about 5, which is maybe not so physiological, although in human body, I think we can find organs, especially intestinal system, where the pH goes down to 2 even. But what we found through the measurement of this zeta potential, as well as the mobility of tubulin and microtubules, that the charge changes from negative. I told you until now that the charge on tubulin is very negative. Now you see that that's true for pH values of 5 or higher, but at lower pH values, it becomes 0 at 5 and positive at below 5. So tubulin becomes positively charged by protonation, basically. The surface of the protein becomes protonated. And we've been able to see this. I don't know if this will play. Yes. We changed the polarity of the solution of the electrodes, and the bundles of microtubules move in the opposite direction, and the same for tubulin. Okay, so there's the effect of sort of electrical polarity on the movement. This is the simulation that was published by a group from Italy and Prague, Czech Republic, 10 years ago, and it shows you this is an atomistic rendition of the electric field around microtubules in mitosis. So here's the mitotic spindle and mitotic spindles in green, and here are the computer simulations of the electric field along the microtubule lines. And these are very substantial fields, 1600 volts per meter along the length, which are stronger than the electric fields in the cytoplasm, typically about 200 to 300 volts per meter, so almost 10 times. And then we finally, more recently, in about 3 years ago, we published a paper that actually explains the mystery of conductivity and capacitance of microtubules by solving the Poisson-Boltzmann equation for microtubule cylindrical objects as a function of temperature and cylindrical geometry and the function of concentration of ions. And these are the results that you can look up the results in detail in that paper, which is free for download. And it shows you that that the local concentration of potassium, positive counter ions, changes as the function of distance to the C termini, distance to the surface of the lumen, and also distance from the surface of a microtubule. And it can be actually the so-called Debye length, which is the length over which a charged object in solution exerts its electric potential, varies from something like 1 nanometer typically at physiological concentrations to hundreds of nanometers at low ionic concentrations. And then this diagram actually illustrates something that I mentioned or hinted at. There's a crossover from high conductivity of microtubules compared to the bulk solution. Solution is red, microtubules with or without C termini are shown here is in green and blue. So the conductivity, as you can see here, can be hundreds of times greater for low concentrations. And then it changes, it goes opposite. Solution becomes more conductive than microtubules. And that crossover point is about 100 millimolar concentration. So the cell could regulate the utility of microtubules as a conductive or capacitive, depending on the ionic concentration. So a cell cycle can do that too, and other things. Maybe in the interest of time, I'll very quickly skip through this. We have a probe, a molecule called P4VB, which binds to microtubules, and when illuminated with laser light at different wavelengths, it emits light at a shifted wavelength, and the amount of shift in the peak of that emitted light is proportional to the pH of the solution. So we were able to quantify it for microtubules and demonstrate that microtubules have a lower pH compared to the bulk solution by about up to 1, which means 10 times greater concentration of positive ions around microtubules compared to the solution itself. That is pH dependent at this rate from 6.5 to 9, and the greater the pH, the greater the difference in these values. So now in the last part of this talk, I will shift gears and move toward time-dependent electromagnetic fields. This is a paper that sort of started this discussion. It wasn't ours. 2017 showed that pulses of electric fields can disrupt microtubule dynamics in human glioblastoma cells. And we started with pulses of electromagnetic fields in the terahertz region, actual low terahertz, single terahertz values, with my collaborators at the University of Alberta, Frank Hegman and his students. And you can see here that a lot of things in cells, especially water, but also proteins, respond to fields in the terahertz region. Terahertz is 10 to the 12th, by the way. It's low frequency compared to visible light and infrared, but has profound effects on the dynamics of microtubules. You see here terahertz off, microtubules in white, very stable structures. When you turn on the field, they disappear over 10 to 30 minutes. And you can see here the breakage of microtubules in this micrograph. And actually that video shows you this dissolving of microtubules in over 15 minutes. It's only we have several screenshots put together to make that movie over 15 minutes. And we also investigated the effects of frequency. So 0.5 terahertz in green is more effective than 1.5 in terahertz in dissolving microtubules. Remember, I told you the resonant frequency was about 0.85. So it's closer to 0.5. That was an estimate, depending on the length. So anyway, but that's in the same order of magnitude. So we're quite happy to see that effect play out. And this is again the lab of Dr. DeGario in Florida, showing that using visible light. In this case, it was 630 nanometers, if I remember correctly, or 660, changes the assembly of microtubules. This beta is the product of number of microtubules times their average length with the electromagnetic field, so light on. You polymerize microtubules in a slower fashion, slowed by between 5 and 10 minutes, and much less is polymerized. And this kind of corresponds to simulations which showed effects of electric fields on the changes in the dipole moment of tubulin and the response of C termini to the orientation, with a critical aspect of it being the strength of the electric field. So it has to be high intensity to do these conformational changes. More recently, we're still digging into it deeper, but Fourier transform in infrared spectroscopy allows you to look at the specific changes in the structure of proteins. Here we have spectra for microtubules in red and tubulin in blue. And very recently, we published work that actually it's not even published, it's submitted actually, which shows you that exposure of tubulin to near infrared in about 810 nanometer wavelength changes the composition of the protein in terms of structural motifs. So there is less, this is control. Exposed tubulin has much less alpha helix content and much more beta sheet content. Beta turn is more or less the same, fewer random coils, which means that exposing microtubules to near infrared affects their conformational state, and that means differences for affinity between tubulins and the rate of assembly. That's the experiment on delayed luminescence, which is 650 nanometer wavelength, illumination of a set of microtubules with 4 minutes separated by 10-minute gap in illumination, and repeated process. And here we see that that delayed luminescence, which is the re-emission of this light from microtubules and tubulin, is a very long process taking place over hundreds of seconds or several minutes. And microtubules have a longer process that to undergo this re-emission. These are more recent, more accurate results, and showing that the half-life of re-emission of photons from microtubules is much longer than tubulin and much longer than the solution itself. Now, and that's the experiment done at Princeton by the group of skulls. This is interesting, and maybe I'll just very briefly explain why this was done. This is tryptophan fluorescence, so you illuminate it with ultraviolet in about 300 nanometer wavelength, tubulin or microtubules, and again make the count of emitted photons. These are quanta of light, high energy light, and we probed that system and obtained delayed re-emission of light over up to 5 nanoseconds. These are nanoseconds, half lifetime of that, which is comparable to photosynthetic systems, even longer, in fact. And for microtubules, it was longer than for tubulin. And we also were able to measure with a very smart way of doing the experiment, the range of correlation of different tryptophans within the system, and obtain something like the correlation range of about 6 nanometers. This is all published recently with the team. Greg Scholes is the principal investigator on this, and 5 nanoseconds. So the idea was to probe quantum effects in microtubules, and we demonstrated they exist, but they are, well, 5 nanoseconds is probably too short for physiological importance. 6 nanometer is roughly the size of a dimer, so each dimer can be seen as a quantum correlated state, but not a microtubule. It's much shorter than a microtubule. Okay, now in the really the last few minutes, I want to switch gears and talk about potential therapeutic importance of all of this. And that started in 2007 with this paper by the group of Kirsan et al. From Israel, and that led to the establishment of the company called Novocure, very much in existence today, producing therapeutic devices, helmets that are used for the treatment of brain cancer, glioblastoma multiforme. They showed that these frequencies of electric fields target microtubules in dividing cells and penetrate into the dividing cells, twist microtubules and spin mitotic spindles, preventing the cells from dividing or delaying the division. So we've done some experiments with these fields. I hope that they will show. This is control HeLa cells dividing properly. No big deal. These are here the same cells dividing in the presence of these fields, and you can see here that there's a great difficulty in separating the cells. The green is microtubule bundles. They still stay connected. So, and the length of cell division is increased by at least 50% in duration. So there are measurable effects on cell culture. We've also calculated from the conductivity properties that electric fields could actually produce local heating comparable to the effects of metabolic energy production. This is the power generated by heating from the effects of electric fields compared to the power generated from metabolic processes. So it's about a third, but that's significant in our opinion. And finally, back to Brazil. This is I'll talk about the use of electromagnetic fields in the treatment of hepatocellular carcinoma, HCC, liver cancer. This is a schematic of the delivery of the field intrabuccal. So in the mouth, there's a antenna, basically a spoon, metal spoon connected to the generative electromagnetic fields. This is how this field is distributed throughout the human body. Power distribution called specific absorption rate in a simulation. And this is the radio frequency electromagnetic field that is being delivered. The carrier frequency is 27.12 megahertz, and the amplitude modulation, the envelope, in other words, changes its frequency depending on the patient from 100, actually even 10 hertz to 100 kilohertz, depending on the response of the patient. So the first, and this is in Sírio-Libanese hospital in Sao Paulo, in Brazil. Fred Costa, Dr. Costa is the pioneer in the use of this therapy, who cured, well, maybe too big a word, but helped many patients live longer with this disease. And so the first is a preparation phase, then there's discovery phase, which basically looks at which frequency, scanning all these frequencies I mentioned to you, and finding which ones the patient responds to in terms of heart rate variability. So it's hemodynamic biomarker for response. And then there's the treatment phase, which is about 1 hour to 2 hours at a time, once a week for 6 weeks or so. And the effects are quite remarkable. These are the histological changes as a result of treatment or exposure to these fields. And then there are also objective responses in terms of tumor reduction, mass reduction, especially in combination with standard therapy, of course. There's a complete response in some cases. So the proposed mechanism of action involves microtubules, which is the connecting theme through this talk, changing the mitotic spindle organization, interacting with electric charges and dipole moments, and then also effects on mitochondria. I have no time to go into the energetics of this process, but there are also experimental data on patients, their quality of life, the amount of energy, metabolic energy produced improve, which we call the reverse Warburg effect, and effects on ion channels, especially calcium, also documented in the literature. So there are currently 3 companies using these types of devices in the therapeutic domain. That's Novocure I mentioned. This is Autem Therapeutics. That's the company based in New Hampshire, USA, but it really is the brainchild of Fred Costa from Sao Paulo. And there's the TheraBionic, which is in Switzerland. So these are the 3 slightly different modalities, but all utilizing electric and electromagnetic fields. And yeah, so in summary, we have started looking at some intriguing properties of biological structures, including microtubules and actin filaments, which I didn't have time to talk about, but we also did that, as well as cells and the human body exposed to electric and electromagnetic fields with some astonishing results. The ultimate goal is to be able to come up with something like this, an equivalent electric circuit for a cell, normal or cancer cell, which also interacts with electromagnetic fields and not just electric potential differences. So I wish to thank you for your attention and apologize if it was too long, but it's a topic that is emerging, and it's becoming, in my opinion, somewhat encouraging for the future of electromagnetic medicine. Thank you very much.
Thank you very much, Jack, for the excellent talk. Very interesting. Thank you very much. Okay, people, now we will start the discussion session. So anyone who has questions, please raise your hand. You can open your microphone and you can ask questions directly. Well, I do have some questions, Jack.
Sure.
For instance, when you talk about brain cancer, okay, you try to use the TT fields into brain cancer treatment. How successful is that? I mean, what there is some measurement that shows that effectively people with brain cancer can live longer by using this particular treatment. There is any statistics on that?
Yes. By the way, this is a very interesting case because as I showed you, in 2007, they published the first basic research paper. And then the company was set up in Israel, I think, called Novocure, maybe 3 years later. And now if you go to the New York Stock Exchange, it's listed there, and it's actually worth a few $1 billion, maybe 7 now, something of this order. So it's very successful financially, but also because of the objective clinical results. So they've done clinical trials in the US and are approved by the FDA. This therapy is also used in Austria, Switzerland, and Israel, and Germany, I believe. And to answer your question directly, these clinical trial results for brain cancer patients indicated something like an extension of life on average between 3 and 6 months. So it doesn't sound like a lot, but it's for brain cancer, it's the life expectancy from diagnosis is maybe 10 to 12 months.
Yeah.
And more importantly, it's non-invasive. and the quality of life of the patients is improved, because the only thing that is required is to wear this helmet with electrodes for about 18 hours a day. So that's inconvenient, but it's nothing compared to brain surgery, you know, or very draconian chemotherapy. So, and currently, I think they are also doing clinical trials for other types of cancer, lung cancer, and I believe also liver cancer. So, so yeah, that's the result, which I think is encouraging. It's not a cure yet, but
Yep.
you know, technology is improving. So that's my answer to your question. It's a very important question, of course, because you need some objective clinical data to validate, and they do have it. I encourage you to go to their website, because there are some patient stories, you know, and some very good results indicated there.
Okay, but I have another doubt as well, because I wonder, okay, you try to treat tumor cells using electromagnetic fields, but what are the effects of electromagnetic-based techniques on non-tumor and immune cells? Do you have some problem with that, in the sense that you can affect also non-tumor cells when you try this particular treatment?
Well, that is a good question too. So there may be some effects, but you know, if the main target is the mitotic spindle, then of course dividing cells, all dividing cells will be affected to some degree. Non-dividing cells or cells in an interface will not be affected much at all. So that's the first positive aspect. Secondly, with the Novocure therapy, it's directional. So the field is directed or focused actually from various electrodes on the tumor. So it's just like radiation can be engineered or designed to have the focal point at the proper target. The case of Autem, the Brazilian/American company, is a bit more complicated because it delivers systemically electromagnetic field to the entire body, but it is focused on highest conductivity areas. Now, the immune system, I would expect this to be somewhat affected, but I don't have any direct information. There may have been some studies recently. I don't know. It's a good question, though, because, of course, you don't want to weaken the immune system of the patient.
Yep. And I have another curiosity, because, okay, you think about brain cancer, you think about those cancers that are very aggressive, but maybe it can be also an useful option, a treatment option for less aggressive cancers, such as skin cancer. What do you think?
Yes, absolutely. And that's the one that would be, in my opinion, the easiest to deliver the radiation to, because that's actually one of the key issues I didn't talk about, but investigated, is the penetration depth of the field. Electric field is exponentially reduced as it penetrates into the body, and electromagnetic fields, that depends largely on the frequency, but also exponentially. So visible light may penetrate over centimeters, but lower field frequencies, even micrometers or millimeters. So with the superficial cancers like skin, of course, that problem is almost non-existent, because all these fields, and you can direct it at will to the presence of the melanoma cells. So yeah, you're right. Although I'm surprised this has not been, in none of these cases that I mentioned, melanoma has been targeted. So it's an open area of investigation and therapy.
Okay, excellent. So we have a question here. Can this technology be used in infectious disease treatment?
Yeah, you see, that goes back to the to my initial slides on the COVID hijacking of microtubules and host cells. I believe so. Maybe so that would require, of course, the investigation of the specific frequencies that would prevent the virus or some other pathogen from interacting with the host cell's cytoskeleton, or even maybe the membrane as well, because they have to get through the membrane. So yes, but I think it's a completely open issue. It's a completely open research program that that would be doable, but in a different institute or different type of facility than.
I see.
I think
okay.
so. I even thought, to be quite honest, I even thought about a slightly different application, namely antibacterial use of electromagnetic fields, destruction of their cell division apparatus, which is different. It's FTSZ rings that, divide bacteria. And if you can find resonant frequencies that these rings would respond to, you could prevent bacteria from dividing.
Okay, nice. Okay, Nicholas has a question. Go ahead. You have to open your microphone.
Hi, Jack. I just have a comment here that is in line with that from Fabrizio. This technology has been used in Belgium for some clinical cases related to arthritis and inflammation. And it seems, and for muscle pain as well, and it seems to alleviate it, so people like it. However, for some reason that is basically stupid, it didn't go ahead, which is contrary to the case from Israel. But anyway, I think it's interesting because it probably has something to do with the human system, as said by Fabrice and so on. So I don't know. Perhaps you can comment on this.
Yeah, I would like to receive information about this Belgian experiment or therapy. Very interesting. I'm not surprised that has been tried, and most of the results that you see in the literature are kind of anecdotal or spotty. I think what one needs to do is systematic. That's what we try to do: explore different frequency ranges, kilohertz, radio frequency, terahertz, visible, infrared, even ultraviolet, and then different modalities respond differently to these frequencies. Now, the question of delivery is another one, and Fabrizio very correctly indicated some easy targets like melanoma, maybe other ones as well, maybe bacterial infections or whatever. So that's the second aspect, and the third aspect is tuning and intensity, sufficient intensity, to have the therapeutic effect. My suggestion to anybody who's serious about it would be basically to start by augmenting the existing standard of care with additional electromagnetic or electrical therapy to improve the efficacy overall. And there may also be resistance to this because of the, I think it's somewhat discredited by some, how shall I put it, witchcraft, scientific witchcraft in the past.
That was exactly what happened in Belgium. It was never taken seriously. And during more than 1 decade or two, it stays in like this without any more development, because they got a patent, but it has never been, it didn't succeed to go through the bioethics stuff, you know, and it hasn't been approved at the end to be delivered at the clinical level.
That's why I think this company in Israel and the one from Brazil are going through proper steps, approval of the devices, safety of the devices, you know, all of this. It takes time and it takes money. So if you don't have sufficient amounts of money, it will not be implemented.
That's it. So thank you.
Thanks, Nicola. And if you could send me some information about this Belgian experiment, I would be very happy to read it.
Okay.
We have two questions, one from the Q&A forum and another one from the chat. I will read the Q&A. You showed how the electric field is capable of tearing microtubules apart. Could this be the mechanism of action of applying the electric field to cancer? What is the reason for the action of the electric field to be restricted in cancer cells? Could it be due to the pH issue?
Well, pH will be one aspect, but I think the main thing is the presence of microtubules in an organized fashion, you know, and forming bundles.
Okay.
Can I excuse myself for a second? I'll be right back. Okay, just give me a minute.
Sure. So everybody, just to let you know that this webinar is being recorded and will be shared on the Global Health Network Latin America Hub. And the link is in the chat.
Okay, I'm back. I'm sorry, but I want to comment on the previous question a bit more, because I think the difference, in addition to the fact, in addition to the fact that you have the main target microtubules assembled and in sort of geometrical organization mode, there's the main thing, in my opinion, is the lower transmembrane potential, which will enable the fields to penetrate, as well as the cleavage furrow. So most of the field lines go through the cleavage furrow. So these two things, I think, the geometry as well as the reduction in the transmembrane potential.
Okay, thank you. We have another question here. Given the great work the research has done to map protein marker charges, it would be theoretically possible to use that data in a LLM to aid targeting of carcinogenic cells? First question. Second question, for the 3 prototypes of the electromagnetic intervention, could you comment on their specificity and sensitivity? Thanks.
Okay, good questions. I think what is LLM?
I don't know, actually.
To use the data in LLM to aid targeting of carcinogenic cells. Well, I think if I understand correctly, that would be like find some electromagnetic markers. I think so. There's a large language model. Oh, I see. So in other words, some artificial intelligence, I presume. So yes, well, that probably is a fairly straightforward task. I mean, it would have to be, let's say, you can easily do studies on hundreds of cell lines, and then use electromagnetic signatures of these cell lines, maybe correlate with aggressiveness or things of this nature. So that's definitely, in my opinion, is a very interesting and important project to be done. Now, the second question is the 3 prototypes. Well, they're not in the same stage of development, so it's hard to compare, but the NovoCure one is the most advanced. I think they've had thousands of patients already. The one from Brazil, Autem, I think it has not been done under proper clinical trial conditions, but they've done maybe 200 patients and analyzed the results. So it's a much smaller study. The third one in Switzerland, I think it's probably preclinical, if I understand correctly. So it's hard to compare them because of the different stages of development. In my opinion, they probably are suitable for different types of cancer. You know, like NovoCure is very well suited for the brain cancer case. And the Autem is more for liver cancer because of the amount of energy distributed. And by the way, there is a very interesting website and a company in Switzerland. If anybody's interested in exploring the effects of electromagnetic fields on cells and human body, different organs, and getting parameters, it's called ITIS from Zurich. And they have a fantastic website with full of data and tables and images of field energy distribution due to different frequencies, the penetration depth. I told you about the various frequencies having different penetration depths. So that is part of the sort of engineering aspect, technical aspect, how to tune it to a particular target, because that's just like drug delivery, targeted drug delivery. You have to tune it to the particular type of disease and cancer location.
Okay, thank you. More questions?
I just want to say that this is my email I posted. If anybody has any questions that are not ready to verbalize here, they can email me with a question and I'll try to answer.
Okay. We have another question here in the Q&A forum. Could the presence of the electromagnetic field help the transport of certain drugs through the microtubes, having in mind its linearity on the electromagnetic field exposure?
I think it's a great question. It's an absolutely fantastic possibility for drug delivery in the future. Combination of natural proteins, natural expressed and forming microtubules. In fact, you can even control their assembly and disassembly by changing pH or ionic concentration. Calcium disassembles, for example. You need zinc for maintaining stability. And then electric fields for the transport of drug molecules on vesicles or through motor proteins. I think it's a great project for the bioengineering of the future. That's not science fiction yet, but it's really a very great, potentially transformative idea, I think, for how the drug delivery can be done. Yeah, I agree.
Okay, great. Any more questions?
And one more thing on this topic. A simpler version of this is so-called electroporation. So use high-intensity electric fields, pulses, which break membranes and cause pores in the membranes, which then allows the drug molecules to penetrate into the cell. And that could be focused on a particular tissue or organ. So that already is in practical existence, I think. But the previous question, I think, was more futuristic, but absolutely doable, in my opinion.
Very good. Okay. This one is okay. Okay, no more questions. Okay, Jack. I think that we can close the session here. Thank you very much, Jack. Thank you for your participation, for accepting our invitation. And I want also to thank you all the attendees for your participation in this webinar. And we will share the recording of this session along with additional materials. We have just posted the link to where you can find these materials in the chat. So thank you very much. Thank you, Jack. Fantastic talk. Very, very good. Thank you so much. And thank you all for participating. We will now close the session. Thank you so much.
Bye-bye.
Bye-bye.